Well, I'm delighted to be here. In fact, I'm thrilled. So I want to just give you a little background of who I am for 2 slides, and then we'll talk about the science. But my world is really science. So I was born in Iowa, and I have 2 daughters, 2 grandsons. I went to the University of Pennsylvania, both for chemistry and medicine. Eventually, I discovered that what was called a slow virus was in fact a protein, and I dubbed these proteins infectious proteins or prions. For that work, I received the Lasker Award in 1994, the Nobel Prize in 1997. And since 1998, I've been working on drugs for Parkinson's and Alzheimer's disease and other related neurodegenerative diseases. So I just tell you a little bit about my private world because it's deeply connected to the Vatican. My partner is a Renaissance art historian, and so we have visited the Vatican many times, and I developed a very special relationship with Joaquin Navarro-Valls, who many of you know, who unfortunately no longer alive. And he opened innumerable doors throughout the Vatican. And it became clear to me that the Vatican is the world's best museum in Renaissance art, without question. And we love travel, go to many different places. But without my partner, I love watching the San Francisco Warriors. So I will stop at that point now and segue into the science. So when I began all of this work in 1972, I've had a patient with Creutzfeldt-Jakob disease that I'll talk about in a moment. And this is what the brain of the patient looked like. So there are all these vacuoles, small vacuoles in some patients, much larger vacuoles in other patients. We don't understand the regulation that takes place that controls vacuole size. And quite frankly, I don't think it's too important to do that. So my work really began in 1972 when I began a neurology residency at the University of California, having now left the University of Pennsylvania, and having spent 3 years at the NIH. I then did a residency in neurology, and it was 3 months into the residency, 2 months, and I had a patient with Creutzfeldt-Jakob disease. And the more I learned about the details, the more I learned that people really didn't know what caused Creutzfeldt-Jakob disease, but it was transmissible. And Gajicek and Gibbs at the NIH showed that CJD and Kuru both would be transmitted into non-human primates by inoculations into the brain and waiting 1 or 2 or 3 years to see illness. Fortunately, there was an animal model that was much smaller, a mouse, that could be used in scrapie, a very similar disease of sheep, similar pathology with all those vacuoles. And so that's where I really began to push my effort into this. And I became captivated. Well, about 4 years later, one could tell for the first time that we began to understand, at least in Creutzfeldt-Jakob disease and scrapie, that if there were no prions present, that in lane 1 was the normal form of the protein, PRP, that we discovered. And in lane 2, if we treated with protease, the protein disappeared completely. And then if the protein was now, we're using antibodies, and we looked for the same thing, but in a scrapie-infected animal's brain, what we found was that there was a bigger signal. And then all the way to the right, we found a protease-resistant form. So now the one that is destroyed in lane 2 is removed in lane 4, and we see what's left is the protease resistance signal, which is the PRP scrapie form. So there are two forms of the protein, the PRPC, the normal one, and from the infected brain, the protease resistant form. We began to look by electron microscopy at all these rod-shaped structures constantly is what we would see. And knowing nothing about electron microscopy, I had the great fortune of meeting Robley Williams, who's shown here. And Robley Williams had just retired. There was mandatory retirement in the world of academia in colleges. And shortly after that, it was overturned in the Congress. But he had a lot of time on his hands, and I could go and talk to him. And he was a fountain of information, and he saw the world the way I did, very quantitatively. So he was the first person to measure the size of a virus using rotary shadowing. He had been an astronomer before becoming a virologist. And so I put these remarks together on the left. I'll read them to you because it's a wonderful story. So Robley, he often remarked, I don't know what these rods are, but we see them in every purified scrapie sample. They don't look like a virus, at least one that I've ever seen. There are no viruses in my atlas that look like these rods, each of which looks slightly different. All polioviruses look the same, but are different from, he didn't say COVID, but I put that in there, from COVID viruses. It's great. All these guys at the virus lab, which was a very famous place, and Wendell Stanley was the one who directed that whole unit at Berkeley. All these guys at the virus lab who thought that I was crazy to spend so much time working with you, now they think you're onto something important. We used to laugh about this a lot. Well, the next experiment we did was to bring George Glenner into this, and I took a sample of purified prions, and by bright field, we could see red with Congo red dye, and then under polarized light, we could see this green gold birefringence, and changed every 45 degrees. So between the cryo-EM, the EM, and now with the, excuse me, the bright field and the polarized light studies, it was clear that this was amyloid. And that opened up the idea that maybe prions were involved in other diseases with amyloid collections. And here you see plaques. Well, this is in Creutzfeldt-Jakob disease, but similar plaques are found in Alzheimer's disease, and they're much more abundant. And so we began to think bigger and more broadly about what might be happening here. And this cartoon, I've acquired this little shape over here on the left, which is meant to be a protein, and the hydrophobic side chains. So 8 out of the 20 residues have hydrophobic side chains, and those are red, and the ones with the hydrophilic side chains, the water-loving ones, are with the blue. Now, there's a conversion of the normal form of the protein to the scrapie form or the prion form. And you see now that the desire of the protein to shove its hydrophobic residues, its side chains, into the internal side of the protein, and then the conversion takes place, and now we end up with 2 of these. And now they tend to form these amyloid fibers because they can push the red residues, the hydrophobic residues inside, and so they can deal with the water on the outside much more efficiently. So in the last almost 60 years to date, you see studies by Michael Kidd in England in a Nature paper in 1963 of what are called paired helical filaments that are amyloid fibers made of the tau protein. And then after a long period of time, this went nowhere in terms of understanding what is going on. But then 3 years ago, I'm sorry, 5 years ago, it was a Nobel Prize in 2017 for the development of cryo-electron microscopy. And this has revolutionized study of these proteins with hydrophobic surfaces. And you can see what these fibers look like. These are tau filaments, and they come from many, many different clinically different diseases. Here's Alzheimer's disease. Here's chronic traumatic encephalopathy. They're slightly different. There are only three residues that switch places. And over here is, progressive supranuclear palsy. So there's a whole category of almost 15 different diseases now that have been examined largely by Soers Shares and Michel Godert at the MRC at Cambridge. And where this all eventually goes is really shown here. And these are now, we can see individual atoms. And this is really a tremendous advance from resolution that did not allow us to do that. Until now, we can begin to see 2 angstrom resolution when we can see individual atoms, and we'll know where they are. And I want to stress that people tried to do this with X-ray crystallography, but the crystals never resembled the prions, because prions don't crystallize. And so now, for the first time, we only need insoluble fibers, like the ones shown all the way on the left over here, and then here in these ribbon diagrams. So this is a whole wonderful, wonderful revolution. So let me just kind of end now and talk about these different diseases in this brief slide, just to give you a sense of how broad this whole area is. So BSE, or new variant Creutzfeldt-Jakob disease, or sporadic Creutzfeldt-Jakob disease, or inherited Creutzfeldt-Jakob disease, these are all the same protein, PRP. And they turn into these protease-resistant forms. And then there's a disease, chronic traumatic encephalopathy, which occurs in people who have repetitive head injuries. And it seems to be this is an area where there's a tremendous amount of interest in this, in terms of, in the past, professional athletes like football players on soccer players, but now there's a tremendous amount of interest in this from all the horrible head injuries from the war in the Ukraine. So there's a lot more to come about all of this, I'm sure. Then down below here is Alzheimer's disease and Down syndrome. And it's become very clear that both in Alzheimer's disease, that it's a double prion disease where the A beta prions form and they stimulate tau to become a prion. And now we know from new studies that are unpublished, but shortly to be published, that in Down syndrome, it's the same story. We have the triplication of the APP gene, and that overexpression, so it's not a mutation, it's just an overexpression of APP gives rise to more A beta, and the A beta then stimulates the tau, and pretty soon we have plaques and tangles in Down syndrome. And then in Parkinson's disease and multiple system atrophy, alpha-synuclein is the key protein that turns into a prion. And this is where, in my own world, we have a tremendous focus on multiple system atrophy, because it is Parkinson's on fast forward, and it's much easier to investigate. We have many more tools. And I have to say that, there's been this tremendous interest in Alzheimer's disease, but the APP gene is very huge. And a large protein. The end product is a very small peptide, but that's probably not the key in trying to develop a therapy, because it's much too late. But in multiple system atrophy, the protein is only 140 amino acids, and there's no alternative splicing. And then there are a lot of functional prions that are listed down below. The work on memory is largely that of Eric Kandel at Columbia, who received a Nobel Prize for much of his work, but not on this particular set of studies. So I think that's the end. Oh, there's one more, maybe. How am I doing on time? So there is a book that I wrote, now old, it's 2014, but it describes all of these early studies and all of the advances that came from these kind of what I would now call primitive approaches. But this opens up a whole new biology. And I want to point out that there are no drugs for any of these diseases. And I think, one of the problems is that this is so new in many cases that it's really been very difficult to convince virologists that some of the diseases they thought were caused by slow viruses are caused by prions, and then to convince a lot of people who don't think that prions are the cause, even though there are these big fibers that I showed you. But I'm hoping that cryo-EM will greatly accelerate the knowledge level and we'll bypass some of the ignorance that seems to be a constant feature of this work. And that's it. I'm happy to answer questions.
Thank you very much. Stanley, is there any question for Dr. Prusiner? Yes, please.
Yes, Stan, I've always wondered about these proteins and to what extent, for instance, with tau, a protein that associates with microtubules, to what extent is the degeneration due to the aggregates themselves interfering with cellular function versus the drawing away of the protein from the microtubules, say, to contribute to microtubule instability, for example?
So I think I'm right, but maybe I'm wrong, that you can knock out tau and you can have a viable animal. And if that's correct, then that's not an issue, because then you can exist without tau. But also, I think that the key is not, there's not a lot of microtubule pathology in people. What there is the accumulation of these prions and then these multimers that form. And so you get all these tangles. And the tangles, I think, are what accumulate in the neurons, and they destroy the function of the neuron. But, nobody knows for sure because we don't have a perfect model of all of this. And, tau is extremely difficult to study because there are 6 different isoforms, and the protein averages about 350 amino acids among the 6 isoforms. So this, and in cells with tau, there are 85 phosphorylation sites. And we don't know the function of phosphorylation, if any, in tau. So we're not sure about this. We don't understand this at all. Tau has been so difficult. So when I stumbled into the transmission of human synuclein prions into transgenic mice, I immediately started to shift a lot of the work into multiple system atrophy. Because this is how people have worked so hard for so long on tau, and they've had so little luck in many cases.
Yes, may I?
Not very encouraging, sorry.
Professor Prusiner, I'm inquiring about the capacity of replication and the environment that's necessary. So you've indicated, of course, the brain tissue, but I'm wondering if there's a model. We heard yesterday about brain organoids. And brain organoids, it was an excellent presentation about brain organoids. Is that an environment in which the prions can be replicating, and then it's a location in which drug can be impacting its replication?
Yeah, so my colleague George Carlson, who was at the McLaughlin lab in Montana for many years and then moved, to San Francisco about 2 years ago to work with us. We tried for many years to develop and with collaboration with Irv Weissman at Stanford, we tried to develop an organoid system, and using chimeric mice, we also tried to do that. And we really couldn't get reproducible data. There are a couple papers in the literature, but we never could reproduce that work. So we thought maybe we had something, but it didn't work out. So I don't know. I think, the problem is the brain is so complex, and there are all of these enormous connections. And they're changing as the external environment increases input into the brain, or we don't know how to do this very well. So yeah, I mean, I got very excited about organoids. I thought this was going to be a tremendous advance, and then we couldn't reproduce it. I'm sorry to tell you.
Okay. There's another question from Dr. Mobridge.
So my question was actually not about organoids, but I think I just wanted to say, I mean, organoids represent the fetal brain, right? So this is like injecting prions into a fetus. I think we should not forget that, and that may explain the somewhat contradictory results of that. Although I do think they might have a certain potential in recapitulating the heritable forms of prion disease like Creutzfeldt-Jakob syndrome and things. But my question actually was, so first of all, thanks a lot, Stan, for a fantastic presentation and for summarizing the field so comprehensively. You summarized all the different types of prions, both the ones that act in Alzheimer or Parkinson, but also the ones that are infectious. And I was always wondering why if we assume such similar mechanisms, why is it that some are infectious, whereas others are not thought to be infectious? Do you think this is simply because we don't eat brains, and because Alzheimer and Parkinson are so human specific and don't occur in the animals that we eat? Or do you think there is something there's an underlying difference between particles that form in Alzheimer and Parkinson, or the ones that form in scrapie and other infectious diseases?
Yeah, I think the amino acid sequences of these proteins are very different, and the ones that are most infectious are the PRP ones, and that's why we've seen more cases, for instance, as you mentioned, iatrogenic CJD. So there we have cases of growth hormone. We have cases of duramater grafts. We have many other examples of this, whereas we don't see that with A beta, and we don't see that with synuclein. So we don't have a lot of examples of this. There are people have found, this is John Collinge's work in England, that you can find some human A beta transmissions at very low levels when you look at growth hormone cases. So it's not only of contamination with CJD, but also with A beta. So, the way these human pituitaries have been collected, now it's all recombinant growth hormone, was to collect batches of about 10,000 of these. So these were from autopsies, and then to purify the growth hormone, because bovine growth hormone has a different sequence. Now, of course, all the growth hormone is made in cultured cells, and we don't have this problem anymore. But when it was happening, the batches of 10,000 there were human CJD prions that were isolated. And so it was about at least one case per 10,000. So if you had 2 or 3 cases, then you got much higher levels. But yes, we're very sensitive in the sense that people got sick. And So it's quite clear these prions have they're different. They have very different amino acid sequences. And so those are the most infectious among humans. And the prions of tau and the prions of A beta and the prions of synuclein are much less infectious. And I think that just has to do with the amino acid sequence.
Question? Yeah.
Stan, I really enjoyed your talk. And I was struck by one example you gave of a non-infectious prion, which is the head injury in football or war. And how does that initiate tau? And is it progressive? And if so, how is it propagated after the injury? What do you know about the nature of this prion?
Well, what we know is that the injury starts and the people are asymptomatic. And it's a repetitive injury from everything we can tell. And there's more data on football and not very much data on soldiers. So we're trying to correct this imbalance now. But the work on head injury is quite clear. It takes a long time. So a lot of these people are they were playing football in their teens and in their early 20s, and then they stop. And 20 years went by, and then they get sick, so they're producing more and more prions in their brains. And that's true of all these diseases. These are not diseases of young people generally. So Creutzfeldt-Jakob disease is a disease of somebody who's 60 years old generally, and Alzheimer's disease is even older. And in Parkinson's disease, they're older people. So I think, there's this age spectrum, and it's very useful in trying to think about these diseases, but it makes it much more difficult to work with them. I don't know, does that answer your question, or have I wandered off here?
Then does it propagate? Is it propagated after the injury to increase in amounts with aging? It suggests that it is in a non-infectious way.
Yeah, well, I think propagation is infectious from cell to cell and within cells. It multiplies.
Okay. Due to the time limitation, there are some things, and Stanley, I can give him a quick roll.